In this lesson, we'll look at the time course of synapse changes during learning, and there will be implications for studying. In previous lessons, we've learned that brains are composed of neurons, and neurons generate electrical signals that travel down the axon to reach synapses on a target neuron. And here we see a magnification of the synapse where there's chemical communication between brain cells. And here we see an illustration of the synapse. And let's imagine this synapse is going to be involved in the learning session. In other words, there will be a communication between the neuron on the left and the one on the right during a particular study session. But prior to the study session, there is some baseline synapse strength. Again, what happens as the electrical signal arrives at the terminal, these vesicles that contain neurotransmitters move to the membrane and fuse with the membrane, thus allowing the neurotransmitters to be released. They quickly travel across the gap, and they hook onto these protein receptors, which are also channels. And when the neurotransmitter binds the receptor, it opens the channel, and charged particles rush into the dendrite. The movement of charged particles is the electrical signal here. And these ions are things like sodium ions that are positively charged atoms. Now, when we proceed with our study session, the scientists argue that the synapses involved or activated during that study session get a short-term increase in strength. And this is happening immediately while we're studying. Now, how does the synapse increase in strength? Well, in this picture, you might have noticed on the membrane of the spine, we have more channels here. So let's compare. Prior to study, and now we're studying. So the target cell has inserted more receptor channels here. So for a given quantity of neurotransmitter released, more channels will be opened, which means more sodium ions can enter, and we'll get a larger electrical signal. So we've increased the strength of communication between these neurons that are activated by the learning session. Now, how can the cell respond so quickly? Scientists think that at these synapse sites on the dendrites, there are spare channels ready to be quickly inserted at the synapse site. So these are like reserves. These are sort of off-duty channels. And when the synapse is activated during a study session, they can be quickly inserted into the spine membrane here to enhance the communication between these two neurons. Now, psychologically, this makes the studied information more easily accessible for a while. So this is like the short-term memory effect, the working memory boost. That information we just previously studied is more easily accessible because the synapses have been temporarily strengthened as a result of that study. Now, we're calling it temporary. This is a short-term increase because just as the target cell can quickly insert extra channels to increase the synaptic communication, so too it can reverse that process. It can take channels out of service. So the idea is that the brain can make quick changes, short-term changes that facilitate, that enhance communication between brain cells that were just activated during some particular learning event, but that these changes are reversible. Now, what about long-term changes? Again, we'll start with the baseline synapse strength. Here's our original synapse. And while the short-term changes involve increasing the number of channels at the spine location there, take a look at the long-term changes that are thought to occur. We actually get growth of the spine itself, and that allows for a more permanent extra set of channels to be inserted into the spine membrane here. So while the short-term increase might have been extra channels, the long-term increase also includes extra channels, but now the spine is larger, so we can have a more permanent set of an increased number of channels in the spine membrane. In addition, the axon terminal, the other half of the synapse, can grow larger and can accommodate more vesicles and more neurotransmitters. So for each signal that comes down to the terminal, more neurotransmitters are released into the synapse. So we've got growth of the synapse. This is a physical growth process that is thought to occur. And these are more permanent changes, more long-lasting changes. But importantly, these changes happen hours later after the study session, and also importantly, during sleep. Now, in previous lessons that we learned that sleep is when the brain undergoes memory consolidation. The hippocampal memory traces are reactivated, and they are sending signals to the cortical memory traces, reactivating those cortical memory traces. Now, why is the brain doing that? Because it's that activity of the cortical memory traces that is going to trigger this growth. So the brain is attempting to make long-term growth here, long-term changes in the synapses that represent the learned information that we are consolidating during sleep. So how is it that brains can do this construction project? Brain cells are living machines that can build new cellular parts called proteins in response to learning events. So when a brain cell is activated by its inputs, it triggers gene expression. So DNA stores the recipes to build proteins. So when a brain cell becomes activated, it starts this construction process. It's going to turn on certain genes so as to build certain proteins. What kinds of proteins? The proteins that can make synapses get stronger. You can think of, bricks and lumber. These are the building materials to make houses and buildings and stuff. Well, the cell has certain proteins that it uses to grow the spines, make them bigger, make the axon terminals larger, insert more receptors, etc. So these are the construction projects that help the cell build stronger synapses. But all of this construction takes time. So this is happening hours after a study session and, importantly, during sleep. Now, interestingly, the newly produced proteins, they have the ability to find just the right synapses that were involved in the learning event. And we can get a better handle on that with this diagram here. Take a look at this target cell. It's got 1, 2, 3, 4 different inputs, so 4 synapses, but only one of them was involved in the learning session. But let's say the target cell did fire. So now the cell is going to turn on some genes, it builds some proteins, and its goal is to strengthen this synapse only. Other synapses that were not involved in the learning session do not get strengthened. And this is important. So the learning-induced changes are specific to the neural pathways involved in the learning. Now let's tell the story one more time from start to finish. So here we have our target neuron, and a learning event has caused some inputs to send signals down to these synapses, and that's enough to drive this target neuron to threshold, and it fires its electrical signal. So we've got 2 activated synapses that are contributing to making this target cell respond. Initially, we're going to get that local temporary increase in synapse strength, but that's a reversible process. That's like the short-term working memory boost. And remember, that's a quick insertion of some channels here, but that process is reversible and will disappear in time. But simultaneously, the target neuron stimulation triggers gene expression that builds materials to strengthen the synapses involved in the learning. So those building materials move out into all the dendrites, but only recently activated spines can use the materials. They have been tagged for synapse growth. So this growth process is what brains are doing during sleep, during memory consolidation. The cortical memory traces are being strengthened by this process of making long-lasting changes to synapses amongst those neurons in the memory trace. Now let's switch up the diagram here and follow the time course of synapse changes using this diagram. Here we have 2 neurons that are going to be involved in a learning session, and we'll say we'll have some baseline synapse strength here, and we're not showing receptors, we're not even showing dendrites here, but the way we're going to model the strength of the synapse is to be looking at how big the terminal is and how many neurotransmitters are being released. So prior to study, there is some baseline strength of this synapse here. Now, while we're studying, we get that temporary increase in synapse strength. And in the previous part of the lesson, we talked about quickly inserting more channels in the target neuron. But here we're just showing the strength has increased by having a bigger terminal and more neurotransmitters being released. So compared to the baseline strength of synaptic communication, we've got an increase in synapse strength here. And this is occurring while we're studying an immediate but temporary phenomenon. Then, because it takes time for genes to be turned on and proteins to be built, and they have to find the activated synapses, after hours or during sleep as well, especially during sleep, we get this physical growth process. These are the long-lasting changes in synapse strength. So again, the spines are going to get bigger, the terminals will get bigger, more neurotransmitters are released. So this is the long-lasting increase in synapse strength. And this process happens well after our study session because the biology of neurons takes time for gene expression, protein construction, and for synapses to actually grow stronger. That takes time. So we can associate this temporary increase, that's the working memory boost, but it's the long-lasting synapse change, that's going to be the long-term memory. And again, that long-lasting change is happening during the consolidation phase of sleep. But how much growth happens during sleep, right? So this is where we've got to test ourselves later the next day. We want to see how much progress did we make in building really strong synapses and really strong memory traces. So during a study session, we get fast, easy retrieval because of this temporary increase. But right during the study session, we cannot measure this effect because we haven't gone to sleep yet. So you see, we don't want to confuse these two. We don't want to mistake this easy access to the information when we're studying for the fact that we have strong memory traces. We haven't had a chance to build those stronger synapses yet. So when we're studying, we must be careful. When we have fast, easy retrieval for information, this is a temporary effect. We haven't done the biological work of making these stronger synapses until we go to sleep. So again, the illusion is we think we know it while studying, but we've got to check our retrieval at a later time to assess the strength of the synapses after consolidation. So we want to monitor the strength of synapses in the long-term memory system after consolidation. And if we fail to retrieve the memory for the information, then we've got to repeat the study session and then self-test again after another delay. So the key to building strong long-term memories is repetition, spacing, and retrieval. So repetition will incrementally build synapse strength. Every time we study the same information, we give the brain a chance to build stronger and stronger synapses. Spacing gives time for consolidation during sleep when the growth occurs. So this is the value of spacing. We're helping our brain do repeated consolidation events during sleep. And that's what's really going to build these strong synapses in our memory traces. And then retrieval after a delay tests the synapse strength in the long-term memory system. So again, we don't, if we do retrieval during a study session, we're going to have the working memory boost, and we might suffer from the illusion that we think we know it. We have to do retrieval practice after a delay to test the strength of the synapses in the long-term system. So there is the study illusion from the point of view of the time course of synapse changes. We don't want to mistake the temporary increase for the permanent increase in synapse strength.